Welcome to this environmental research seminar. This is sponsored by the Michigan Life Stage Environmental Exposures and Disease Center and Integrated Health Science Core. And I am Sungkyun Park. So I am very happy to introduce today's speaker, Dr. Sean Harris. So Dr. Harris is a research assistant professor of environmental health sciences here at University of Michigan School of Public Health. Dr. Harris earned his PhD in environmental toxicology from the University of Washington, where he conducted his dissertation research in male reproductive toxicology. So as a part of his research, he helped to develop an in vitro tested cell culture model for studying male reproductive toxicants. He now studies female reproductive toxicology using a combination of approaches, including primary tissue culture models, transcriptomics, and metabolomics, to investigate molecular mechanisms of toxicant effects on the placenta and fetal membranes. His research aims to advance our understanding of how environmental toxicants contribute to adverse pregnancy outcomes. So before I turn this over to Dr. Harris, so while Dr. Harris is presenting, so if you have any question, please leave your question to the chat box, and after his presentation, so he will respond to your question. So Sean, it's your turn.
Thank you. And want to just say thank you to everyone for being on the call today. So I'll just jump right into it. So intrauterine bacterial infections during pregnancy can lead to a number of adverse pregnancy outcomes, including miscarriage, neonatal or maternal sepsis, as well as preterm birth. In addition, research over the past few decades has shown that certain chemicals that we're exposed to in the environment can interfere with the immune system, potentially interfering with the body's capacity to fight off pathogenic infections. However, relatively less research has been done studying these types of phenomena in the specific context of pregnancy. So as female reproductive toxicologists, we investigate how chemicals in the environment may impact some of the specific tissues and organs responsible for maintaining a healthy pregnancy under conditions of intrauterine infections with pathogenic bacteria. So toxicant pathogen interactions typically are, when we're thinking about that, we're thinking about chemicals interfering with the immune system. And as I said, sort of interfering with the body's capacity to fight off a pathogenic infection. And the research that's been done in immunotoxicity, kind of at its most simple level, we've seen chemicals in the environment that we're exposed to can either suppress immune responses or in some cases overstimulate immune responses to pathogens. So we see multiple sort of mechanisms of toxicity, but that can take different forms. Today, I'm going to be more focusing on the idea of immunosuppression or suppressing immune responses to pathogens. The chemical we study is called trichloroethylene. Trichloroethylene is an immunotoxicant, and it's generated public health concern for a few different reasons. Trichloroethylene is a volatile organic solvent. It's used in various industrial processes. It's used in making refrigerants and manufacturing refrigerators. It's an important chemical intermediate for some of those processes. It's also used as a metal degreaser. So workers that are working with machine parts sometimes use trichloroethylene to get grease off of machine parts. Because of its widespread usage, there are many millions of pounds of trichloroethylene disposed of or released into the environment every year. So for instance, in 2015, that was around 2 million pounds were disposed or released into the environment. And for this reason, it's a common environmental contaminant found in groundwater samples, drinking water supplies. It can be released into the air, and people and exposures near some of these contaminated sites can be a little higher due to inhaling this volatile chemical. It's found at contaminated waste sites. You see some of the highest levels of exposure in occupational settings. And so exposure to workers working with trichloroethylene is, kind of concern. So importantly, trichloroethylene can disrupt the ability of the body to fight pathogens. So for example, some animal studies have shown, this is a study done in 2010 by Selgrade and colleagues, in which they exposed mice to trichloroethylene and then instilled the mice with a pathogenic bacteria into their lungs. And then they basically quantified the amount of bacteria remaining in the lungs, which is shown on the y-axis here, at various time points after the installation of the bacteria. And so the mice that were breathing in control air, normal air with no trichloroethylene, were able to clear a lot of this bacteria out of their lungs, as shown by a much reduced amount of bacteria remaining in the lungs after about 24 hours. However, the mice that were breathing in trichloroethylene at range concentrations from 50 to 200 parts per million had a much higher level of bacteria remaining in the lungs at the same time point. It's been found that trichloroethylene metabolites, as opposed to trichloroethylene itself, are responsible for at least some of trichloroethylene's immunotoxic effects. There are multiple metabolic pathways of trichloroethylene, so the way our body metabolizes trichloroethylene into various metabolites of trichloroethylene. One of the major ones is the glutathione conjugation pathway, which gives rise to a toxic metabolite of trichloroethylene. So this is a small snippet of the glutathione metabolic pathway, showing conjugation with glutathione and then this toxic metabolite called S12-dichlorovinyl L-cysteine, or DCVC, which is what I'll call it for the rest of this talk. So DCVC has been shown to be responsible for some of the kidney toxicity that we've seen with trichloroethylene exposure. In our lab, relevant to female reproductive health, we've seen cytotoxic effects and other effects in placental cells with this metabolite. And importantly, we've seen suppression of immune responses to bacterial pathogens in fetal membrane tissue. So the fetal membranes are an important part of both as an important component in defense against pathogenic infections during pregnancy. And they're also really critical to maintaining a healthy pregnancy. It's maybe not a tissue that we think about a lot, but the fetal membranes are like pretty biologically active and very important. So the fetal membranes are composed of two layers, one called the amnion, which directly surrounds the fetus and contains the amniotic fluid. And then the next layer out, sort of going towards the maternal side, is the chorion, which is basically an extension of the placenta that surrounds the amnion. So these two layers together are what we call the fetal membranes. The membranes rupture near term, so they eventually weaken when labor is imminent. And when the membranes weaken and then finally rupture, they expel the amniotic fluid. And this is what we know of as the water breaking. But they can rupture prematurely, which is what we call premature preterm rupture of the fetal membranes. And this can be associated with numerous health problems, including preterm labor and other maternal and fetal health problems. Intrauterine infections, importantly, can cause preterm membrane rupture due to activating inflammation and other signaling pathways that lead to the weakening of the membranes. So the way intrauterine infections work is bacteria colonize the vagina and cervix and then migrate into the uterine cavity and potentially into the fetal membranes themselves. This is what we call an ascending infection. If bacteria cross the fetal membranes, they can then end up in the amniotic fluid, potentially colonizing and infecting the fetus. And this infection causes adverse neonatal or maternal health problems or adverse pregnancy outcomes like those I listed before: miscarriage, premature rupture of the membranes, neonatal or maternal sepsis, among others. part of the innate immune response to pathogenic bacteria is the upregulation of certain cytokines such as TNF-alpha, IL-1 beta, and IL-8. And so these cytokines help to mediate the immune response to the pathogen by, they have a number of roles to play in this response, but these include things like immune cell recruitment, signaling for immune cells to come to the gestational tissues to help fight the infection, as well as activating macrophage antimicrobial activities. So macrophages are the important immune cells that are contained in gestational tissues that fight and basically swallow up and kill pathogens. One pathogen that presents a particular problem during pregnancy is Group B streptococcus. Group B streptococcus can be found both in the gastrointestinal and genitourinary tracts. It's not all that uncommon. About 1 in 4 pregnant women carry Group B streptococcus. That doesn't necessarily mean it's going to become an invasive or ascending infection situation, but they are present. It can just be present and not lead to this more severe situation with an ascending infection. But in some cases, it can transition from an asymptomatic to invasive or pathogenic situation. So we study the biology of the fetal membranes due to its important role in dealing with pathogenic infections. And what we use what's called a fetal membrane explant model, in which we obtain placental tissue from the hospital, from planned cesareans before labor has started. And these placentas have the fetal membranes attached. So we bring them back to the lab and we generate we generate these fetal membrane explants using a pretty simple biopsy tool. And we generate these full thickness punches of the fetal membrane tissue. We then attach these full thickness punches with both the chorion and amnion layers to what's called a transwell insert, which is a piece of plastic that inserts into a standard tissue culture dish or tissue culture well. And so this forms an approximation of both the chorionic side and the amniotic side of the fetal membranes. So a former student in our lab who's now an assistant professor at Calvin University, Erica Boldenau, saw some very striking results with this trichloroethylene metabolite, DCVC, in this fetal membrane model. So what she saw was when her and colleagues working on these experiments treated the explants with this metabolite, DCVC, and measured levels of cytokine release from the fetal membranes. They saw that DCVC on its own, so these are different concentrations of DCVC ranging from 0 to 10 micromolar. DCVC on its own was not doing much. However, Group B streptococcus, kind of not surprisingly, induced a pretty strong inflammatory response to in the tissue. However, when she co-treated with DCVC, the fetal membranes with DCVC, there was a strong inhibition of this increase in this pro-inflammatory cytokine. And she saw this across several other cytokines as well, including IL-1 beta and IL-8. So kind of the same pattern of strong response to Group B strep and then a really strong inhibition of that response with DCVC treatment. So this is really striking data. However, there's a lot of questions we still had after observing this data. Many of the host factors that lead to the Group B streptococcus invasion into the uterus and fetal membranes are not well understood. So how the body is responding to Group B strep and how Group B strep circumvents some of the natural defenses against infection are really not understood and are under active investigation. So there's still a lot we do not understand. So one of those is we wanted to know what are the broader set of molecular pathways activated by Group B strep in the fetal membranes. And then more specific, kind of more relevant to the specific health outcomes that we're interested in, what are the potential implications for adverse pregnancy outcomes related to these molecular pathways that are activated by Group B strep? So as a follow-up experiment, we generated these fetal membrane explants, as I demonstrated earlier, and then treated the explants with group B strep for various time points, so from 4, 8, and 24 hours, in order to look at what molecular pathways were being impacted across, the first, essentially the day of infection with this organism. So then we extracted RNA from the explants and conducted transcriptomic analysis using microarrays to answer this question. So this is the data across the 3 time points at 4, 8, and 24 hours. And you have upregulated genes in red and downregulated genes as the green bars. And so what we observed was that the 4-hour time point, there's about 45 genes that were initially upregulated 4 hours after inoculation with the group B strep. That grew to a little over 100 at 8 hours, and then kept climbing at the 24 hours to about 200 genes were all significantly upregulated in response to group B strep. A little bit later, there was this, we started to see downregulation of a set of genes that became, most pronounced at 24 hours. So a pretty robust gene expression response to this pathogen. So of course, we were interested in not just the number of genes, but the functions of these genes. So we did a pathway enrichment analysis using software called LRPath and enrichment testing software. Basically looking to see which biological pathways were overrepresented amongst the genes that were, in this case, upregulated in response to group B strep. And as we had hypothesized, we were seeing pathways such as inflammation and pathways related to cytokine signaling or cytokine production were enriched amongst the upregulated genes in response to this pathogen. What was pretty cool, though, was we were able to pick up a couple of pretty more specific pathways, such as premature birth and genes related to premature rupture of the fetal membranes were also, these pathways were also upregulated in response to group B strep as well. So that told us that there's a molecular response here that is characteristic of some of these health outcomes that we're directly interested in. So that was pretty cool to be able to get that level of specificity out of this in vitro model. So this is kind of a bit busy figure, but what this is we conducted what's called a network analysis. And essentially what this is a curation of known protein-protein interactions amongst genes within a particular pathway. So in this case, we were focused on the inflammation, the genes involved in the inflammation pathway. And the green arrows indicate activation, so genes that have some kind of activation relationship with one another. So there's a lot of activation amongst the genes in this inflammation pathway. So one thing this allows us to do is identify highly interactive nodes, or the genes within this pathway that have a lot of interactions with other genes, indicating a potentially really responsible role in the overall regulation of that pathway. And so we saw that the gene that ranked the highest in terms of the amount of interactions was TNF alpha, which was upregulated at that 4-hour time point after the inoculation with group B strep. So that had the highest number of interactions and was also activated upregulated early in response to GBS or group B strep. In addition, we saw IL-1 beta, another pro-inflammatory cytokine, was ranked pretty high in terms of the number of interactions. And that was also upregulated at this early time point. IL-1/8 as well was a highly sort of interactive node, if you will, within the network. And that was upregulated not at 4 hours, but at 8 hours. So what was really striking was these were all the cytokines that were inhibited by DCVC. So DCVC is apparently suppressing or inhibiting the expression of these cytokines that are key hubs in this inflammatory response network, or at least highly interactive hubs within this network. So the fetal membrane model, explant model, is really cool. It captures the architecture of the fetal membranes, as shown here in cross section, with both the chorion and amnion. However, because it is full thickness tissue, it contains a variety of cell types. So these include all the cell types that make up the fetal membranes, like epithelial cells, fibroblasts, as well as resident macrophages are found in this tissue. And so we were interested in identifying if there was a certain cell type within in the fetal membrane tissue that was being targeted by DCVC versus the other cell types, or is it multiple cell types? We still had a few, we still have multiple questions about what exactly is happening here. So we decided to focus on the resident macrophages as a potential target because resident macrophages are critical to immune responses. So this critical immune cell type is found in many organs and tissues. And so our goal is to identify DCVC impacts on inflammatory pathways in macrophage cells. So we used a macrophage cell line called THP1, and in this case, we used an activator, a known molecular activator of inflammation called lipopolysaccharide, which is a component of bacterial cell walls. And this was just our way of activating inflammation pathways and then seeing how DCVC modified this inflammation response. So we had 4 treatment groups with these macrophage cells. We had a control DCVC treated group, a lipopolysaccharide treated group, or a DCVC plus lipopolysaccharide treated group. So then after each of these treatments, we isolated RNA and collected cell culture media. The RNA was used for RNA sequencing to conduct transcriptomic profiling, and the cell media was used to quantify release of pro-inflammatory cytokines by the cells under each of these conditions. Our main point of comparison for this particular experiment was between these last 2 treatment groups, lipopolysaccharide treated versus DCVC plus lipopolysaccharide, to see how DCVC was modifying, again, how these inflammatory responses in these cells. So jumping into the RNA sequencing data, these are volcano plots that just show as the red dots indicate significantly changed genes under different comparisons. Based on we applied a fold change, so the fold change is on the x-axis and the negative log of the p-value is on the y-axis. So we applied both of those criteria for our significance criteria. So we saw each condition compared to control, DCVC treated, lipopolysaccharide treated, or the co-treatment, when compared to non-treated cells, induced a fairly robust set of gene expression changes from around 800 to about 2,000 genes, significantly changed compared to non-treated cells. However, as I said, the focus of our comparison for these purposes was between the DCVC plus lipopolysaccharide versus lipopolysaccharide alone treated groups. And so we indeed did see a robust set of gene expression changes there too, with about 1,400 genes significantly changed with the DCVC co-treatment compared to lipopolysaccharide alone. So again, we conducted a pathway enrichment analysis to see in the DCVC plus lipopolysaccharide co-treatments versus lipopolysaccharide alone. So we looked at genes that were downregulated in the DCVC treated group and looked at pathways that were enriched amongst the genes that were downregulated, and saw, as we had hypothesized, pathways related to inflammatory response, cytokine production, cytokine signaling were all impacted by DCVC compared to lipopolysaccharide treatment alone. So again, at the pathway level, we're seeing that DCVC is doing something to really downregulate genes involved in these inflammatory responses. And in addition, at the level of actual pro-inflammatory cytokine release from the macrophages, that was impacted as well. So again, this is that familiar pattern where when the macrophages were treated with DCVC at several concentrations from 1 to 10 micromolar, there wasn't much going on. Lipopolysaccharide or LPS stimulated a pretty robust, significant increase in pro-inflammatory, in this case, IL-1 beta, into the cell media. And then when we co-treated with DCVC plus LPS, we saw a pretty clear dose response in which there was an inhibition of this pro-inflammatory stimulation. We saw this across several cytokines, including IL-6, as well as TNF-alpha, with this fairly nice dose responsive inhibition of inflammatory cytokine release. So going back to the pathway data, so we saw these pathways related to inflammatory responses. However, we were also interested in understanding a little bit more about how DCVC might be impacting specific functions of macrophages beyond just these inflammatory pathways. So we queried and looked into certain pathways such as phagocytosis and cell killing, which are functions of macrophages. They phagocytose, basically engulf pathogens and then release certain chemicals that kill pathogens as part of the immune response. And we saw indeed that pathways related to phagocytosis and cell killing were also enriched amongst the downregulated genes with this DCVC treatment. So that was pretty cool to be able to see this sort of specific set of functions impacted with DCVC as well. So this was allowing us to see something a little bit more specific beyond just inflammation pathways. So yeah, so just as an illustration of phagocytosis, so this is a nice illustration of how macrophages help to deal with bacterial infections. You have the macrophage moving towards bacteria, engulfing the bacteria, containing it within a vesicle, and then releasing what are called lysosomes, which basically have like lysis, contain like lytic enzymes that break down and digest the bacteria. So this is obviously an important function of macrophages, and we wanted to look at this function as well, and to see how this was responding under conditions of DCVC treatment. And there's a pretty cool assay that you can do in which you can obtain bacteria that are labeled with a fluorescent tag that is sensitive to pH changes. So when the bacteria are outside of the cell at a fairly neutral pH, not much is going on. However, when phagocytosis occurs and the bacteria are engulfed in these vesicles that are at a different pH, they're at a much lower, more acidic pH, it triggers a fluorescence reaction, which you can then quantify using a standard sort of plate reader. And so we looked at phagocytosis as indicated by relative fluorescence units under conditions of treatment with DCVC. So you can see here, so this is like relative fluorescence units as a measure of phagocytosis, and then at various time points. The control cells that were not treated with DCVC had this sort of baseline level of phagocytosis. The 1 micromolar of DCVC there was maybe a little bit of an inhibition, but it seemed to catch up, after a few hours. The 5 micromolar DCVC, we started to see a bit of inhibition, but then at the 10 micromolar, we saw this very, this significant across multiple time points inhibition of phagocytosis with that concentration. So as a summary of what I've talked about, in our first experiment, we confirmed that DCVC was suppressing expression of key hubs in this inflammatory response network, using and also that these hubs were part of the early response to group B strep. In the second experiment with macrophages, we saw that DCVC was under conditions of inflammatory stimulation. We saw that DCVC was downregulating genes involved in multiple inflammation processes, as well as macrophage functions, as well as pro-inflammatory cytokine release. And then finally, in the last experiment, we saw that there was an inhibition of actual phagocytosis with increasing concentrations of DCVC. So for conclusions and future directions, our conclusions are DCVC suppresses pro-inflammatory responses and pathways in macrophages. And these findings suggest that DCVC impacts on macrophages could explain at least some of the immunosuppressive effects we've observed in the fetal membrane tissue. So these have potentially important implications for potential hazards associated with trichloroethylene exposure. For example, if immunosuppression in these tissues during pregnancy leads to an increased susceptibility to pathogenic infection. Potentially, this could lead to increased susceptibility to a variety of adverse pregnancy health outcomes, like I said, miscarriage, premature rupture of the membranes, maternal and fetal, neonatal and fetal effects as well. So for future directions, we want to obviously continue to investigate the mechanisms of DCVC immunotoxicity, hopefully moving into some in vivo models, conducting a few more mechanistic experiments to really understand what all DCVC is doing in the macrophages and in the fetal membranes as well, and potentially in other, macrophages are far from the only immune cell, involved in the response to intrauterine pathogenic infection. So there's still a lot more to this story, but we've seen some very interesting results that we want to follow up on and really understand what DCVC is doing. Need to move some of my stuff out of the way here. In addition, we want to use these approaches and these models to evaluate additional chemicals and pathogens. There's a variety of chemicals that are relevant for human exposures that have been shown to impact the immune response and interfere with responses to pathogens. And there's a variety of pathogens that are relevant to adverse pregnancy outcomes, urea plasma species and various other species of bacteria. So we want to continue to understand how chemical exposures may be contributing to impacts of pathogens during this very specific and sensitive life stage that is pregnancy. You have certain tissues and certain organs that are relevant to pregnancy that are not relevant to really any other life stage. And so and relatively little research compared to some other types of research has been done in these specific tissues and organs. So I think it definitely bears further investigation. So with that, I'd like to thank the funding sources that have supported me while I've been doing this research, and my various collaborators, members of the Locke Caruso Harris Lab, who I've worked with, especially Dr. Rita Locke Caruso, who has been instrumental in mentorship and guiding me as I've conducted this research. Dr. Erica Boldenau, who is a continued collaborator, and we're really excited about some of the stuff that we're putting together to continue to address some of these questions that I talked about. She's an assistant professor at Calvin University, and yeah, just a really great and fun collaborator. Dr. Kelly Mikulski and her lab have been super helpful in guiding us through some of the bioinformatics analysis we've conducted. Dr. Mark Chames at Medicine, Obstetrics and Gynecology has been instrumental in helping us in our interfacing with the hospital in acquiring the placental and fetal tissues. And Dr. G here in the environmental health department has been super helpful and instrumental in guiding us through some of the experiments involving microbes and pathogens. So with that, I will take any questions and open up so I can actually see people's.
Yeah. So great presentation. Thank you. So let's see. So I don't see any question in the chat box, but so if you have any question, then maybe you can just raise your hand, or still you can leave your question in the chat box. And so we have about a little more than 10 minutes. So while people are thinking about their questions, so let me ask one question. So this trichloroethylene in general in pregnant women, so can you talk a little bit more about the general like a scenario of exposure, and then your study mainly looking at the acute short-term exposure, I guess. So have you ever thought about just long-term low-level exposure, and then how that could be different from what you see in your experimental setting?
Yeah, I think that's a great point. Our exposures in trichloroethylene kinetics and metabolism, or some of the metabolites are kind of tricky, especially DCVC. It's not especially stable, so getting really good levels it's not the easiest thing to do, but based on the levels of other metabolites in the metabolic pathway that are kind of close in the metabolic pathway to DCVC, our concentrations are probably the best approximation would be on the high end of an occupational exposure, just to give a sense of the relative concentrations we're working with. But yes, I think, levels that you're going to encounter in, like, say, drinking water supplies or in the air, in ambient air, there's not a ton of it in the ambient air in most places. However, close to some waste sites, you can get higher levels in the air, like 50 parts per million or billion, sorry, which that could end up, that would get you kind of closer to the levels that we're looking at. But I definitely think longer term, I'm all about, kind of pushing the models and developing an exposure that really kind of reflects real world exposure. I think, exposure in contaminated drinking water over several days, I think that would be definitely worth looking into, because, our time points are like 24 hours. We haven't pushed it much further than that. We're seeing effects early in some cases, as early as 4 hours. But I think definitely we should look at lower levels and really get the time and dose dynamics correct, because that's going to be really important for risk assessment and hazard identification. But I think that you make a great point.
Thank you. Yeah. So anyone has any question?
Hi, Sean. This is Jackie.
Hi,
Great
Jackie.
talk. I love to see where your research has gone. I'm wondering if you have plans to use this really neat model for, reproductive immunotoxicity for other chemicals.
Yes, we're talking about that. We want to, I've been interested in, we've seen some immunosuppressive effects with PFAS. There are other chemicals that are kind of structurally related to trichloroethylene, like tetrachloroethylene. It's used in a lot of dry cleaning facilities. Dry cleaning workers are exposed to them sometimes. And so I think that would be definitely one we need to follow up on. We've done a little bit of kind of preliminary stuff with perchloroethylene metabolites, but I think that one's definitely one to follow up on. Additionally, there are things like heavy metals can interfere with immune processes. That would be another one I'd want to look at. And toxicology is really moving towards this mixtures model. So I don't know, where we'll go with that. But I think, again, going back to developing a more relevant model, I think we'll probably have to take a look at mixtures at some point. So it wouldn't take a lot of, say, additive interactions between two chemicals to really kind of push the threshold of some of these effects into kind of concerning territory in terms of human exposures. So I think it really does bear looking into, both on the time basis, the dose basis, and potentially multiple factors sort of basis. So we've got two factors here with a chemical and a pathogen or activation of inflammation, but there are other factors that might be important to consider as well.
Great. Any other question?
I have a comment. It's Gil.
Go ahead.
Hi, Gil.
About 40 years ago, I was a reviewer of an article in the Journal of American Medical Association about rare and lung infections. And it turned out at the CDC, where they have a lab that gets samples from all over the world, that all of these unusual cases occurred in the background of silicosis in the patient. So they asked me to write a commentary. What I wrote about was about the interaction of toxicology and infectious disease. That's say chemical exposures and infectious exposures, and how this has to be a very common underlying mixtures phenomenon. I've never seen it really worked up substantially, Sean. And your comment just now about not just chemical mixtures, combined exposures, but chemical and infectious or pathogen interactions really is an area calling for a serious address. So I encourage you to look into it and really think about how to develop it, how to even think about a risk assessment framework for it. And there are now so many more databases than there were 40 years ago. And I think you can make quite a few projects out of this. And of course, the endpoint is very important.
Yeah, absolutely. Thank you. Yeah, and I think, even this has potential implications even beyond pregnancy. So we're seeing effects in macrophages, which obviously are found all over the body and play such a wide variety of, roles in biological processes. So, I don't want to necessarily, we focus on pregnancy, but I think, it's definitely something that should be explored further in multiple contexts. Absolutely.
Pregnancy is particularly salient. I wouldn't go away from that. I would think exploit it.
Yeah, no, I was just saying at the top of when I was talking earlier that I've caught the pregnancy bug. I think the biology is just completely fascinating. So I'm not going anywhere in that regard.
Good.
Great. Yeah. Maybe I want to have a follow-up question. So this group B Streptococcus is actually I'm not very familiar with this pathogen, but I just guess that you look at specifically this is because this is very prevalent in pregnant women, and so that's why you focus on this. But is there any other pathogen you consider that could be important in terms of this interaction with the trichloroethylene, and also any potential interaction with the virus, not just bacteria, but virus and this chemical exposure?
Yeah, there's several species of bacteria that are potentially relevant. Urea plasma species. I'm not a microbiology pathogen person, but so I don't know all the species by heart or anything. But urea plasma is another one. E. coli infection can be an issue in pregnancy. There's several others that unfortunately aren't coming to mind right now. But I think, and then viruses too. I've been very, obviously viruses are on everyone's mind, and that's definitely another one I would love to look into. I feel like I have a lot on my plate, even like understanding all the as a toxicology guy, understanding all the bacterial biology. So I'd want to work with like a virologist. But I think, definitely, it's like I said, the relevancy of any of these to public health is like enormous. So I really want to like push it as far as we can in terms of capturing this stuff.
Yeah, thank you. So any other question? Oh, so we have a question in the chat box. So do you see the comment or question in the chat box?
I might have to stop sharing to see that.
Yeah. Well, I can read.
Oh, yes. No, I can see this is great. Oh, I guess I should read it so people, in case people aren't.
Right.
Is there any other way that the TC metabolite affect the placenta? I believe you mentioned in Dr. Colasino's class that the placenta is a high energy organ. Is it possible for the TC metabolite to also affect the energy consumption of the placenta? And yes, absolutely. We have, one hypothesis I want to explore. We have several publications that Ilana Elkin, who's now a postdoc in our lab, has authored, looking at the effects of DCVC in, these are, I don't want to get too in details about which specific cell type, but cell types in the placenta, and impacts on energy consumption, energy metabolism, and mitochondrial function. And mitochondria are actually really important in activating the inflammasome response. So I'm hypothesizing that potentially that something that explains both of those types of effects could be rooted in DCVC impacts on, specifically the mitochondria. I don't know if that's the case or not, but yes, it does affect energy consumption and energy metabolism in the placenta as well. It's also cytotoxic. It triggers apoptosis pathways, which are tied to mitochondrial signaling. So you're basically right on the money there with that.
Great. So so this is 12:48. So I think so. This is a good presentation, and then I appreciate so your excellent presentation. And before we end this session, so I wanna remind you that so the next week is the M-LEEaD Environmental Statistics Week, and then so we will have a 1 hour session so throughout the week. So so from Monday through Friday at noon to 1 p.m. So there will be various environmental statistic related topic, and so please we will send announcement again, and so please stay tuned, and hopefully we can see you all in that session as well. So thank you very much, and take care. Bye-bye.
Bye. Thank you.